All right, so if you are viewing this, I'm beginning it here because the recording wasn't on. And so let's see how many slides you've missed. You've missed 5 slides. I just talked about the 6th slide here, and you'll have to learn this material on your own. All right, let's move on. Talking further about the viral structure, surrounding the viral genome, the internal most molecule of a virus, there will be a coating protein around the genome of the virus, and that we call the capsid.
Can you share your screen now
Oh,
for the slides?
not doing too well today. Thank you for your assistance. All right, now you can see it.
Yes, thank you.
All right, so around the genome we have the capsid, that's the protein coat, and all viruses have a genome and a capsid, the protein coat. The protein coat is composed of one or more proteins. This virus here, it's just repeating units that we call capsomeres of one protein, and it just repeats. Let's blow that up a little bit. Let's just do it here. So on this virus here, you can see each of these is one protein, and there's a lot of proteins to make the protein coat. That was a little bit slow on focusing, but let's bring it down. Outside of the protein coat, some viruses, but not all viruses, have an envelope. An envelope is a lipid bilayer. It does have proteins and carbohydrates in it, but essentially it's mainly phospholipids. And we'll talk a little bit more about the envelope later in this course. But the point is, the envelope is outside of the protein coat of some viruses, but not all viruses have an envelope. External to the envelope in some viruses is the glycoprotein spikes. Let me scroll down a little bit. Whoops, too far. Pull it up a little. So this is showing you the protein spikes on the envelope virus, and you only find the protein spikes on an enveloped virus, meaning these spikes inside the envelope. These spikes serve as antigens, meaning these are the points that the host recognizes the virus as being foreign, and an antigen is just something that the host recognizes as being foreign to the host, and so it can mount an immune response against these spikes. Other viruses, if they're missing the spikes, the immune system will have to find something on the outside of the virus to recognize as an antigen. And antigens, all they are is a molecule on the surface of a virus or other invading microorganism that the body recognizes as foreign to the self. That's just what an antigen is. Also, these spikes are what the virus uses for attachment. I briefly mentioned that the attachment molecules on the virus are the outermost molecules of the virus. So where are the attachment molecules on this virus here? Okay, this virus here, where are the attachment molecules on this virus? The attachment molecules are what the virus uses for attaching to the host cell.
The points?
That's a really good guess, and you are correct. It's actually since the points are made out of the same capsomere, meaning the same protein, it's every protein of the protein coat, the capsomere, or the capsid, is the attachment point. So this virus uses this capsid protein for binding to the host cell. Any questions about that? And yes, actually the points are the furthest out, so they would be the most likely, the first part of the virus that would attach to the host cell. Let's shrink that a little. About there. All right, also talking about viral structure, we talk about the general morphology of viruses, and general morphology is the shape. You should know this word from when we were talking about bacteria. Morphology also means the shape of the bacteria. Viruses may be classified on the basis of their capsid structure. And there are 3 basic viral shapes. So that's similar to prokaryotic cell. There are 3 basic viral shapes, although there are exceptions. In the viruses, there are no exceptions for the 3 viral shapes, although different authors can have different numbers. But we'll say it with our author, and he uses 3, and most authors do. A helical virus, where the capsid is in a helix, and here's a good example. You can easily see that this is a helix. The polyhedron, where the virus has many sides, and that would be like the complex dice that have many sides, the polyhedral dice that some people play with. And there's an actual polyhedral virus seen under the electron microscope. And then the third category of viruses is just complex viruses, and they can take on any shape and essentially any size. Obviously they're small. Here's a complex virus. This one is an envelope complex virus, and I don't know about this one. It's not labeled and I can't read that. Let me blow it up. It doesn't matter if it's envelope or not, but there's a complex shape, and it sort of looks egg-like. I don't even know what shape you'd say that one is. And then this one looks like the Apollo lunar lander. Okay, very complex shape, and that's bacteriophage T4, which we saw in the video. Any question about that? You guys are probably so young you're not even familiar with the lunar lander from the Apollo missions. Hopefully you've seen it on television or on some museum or something.
Saturn V.
Sorry?
Saturn V. It was the rocket that took up the lunar lander.
Okay, good. That's an old movie. So when we're talking about bacteriophage T4, it's in a class of viruses that we call the T-even virus, and that's because they're named T4, T4, and T6. We'll really only talk about bacteriophage T4, but they all take this shape. And you can see it's a complex virus. It does have DNA for its viral genome, and then it's surrounded by a capsid, but it's more than one protein. There's different proteins in the head of the capsid than in the tail sheath, and then different proteins in the tail fiber, and we won't talk about the pin and the base plate, but that's part of the tail. In fact, all of this portion here is the tail. The head is only this portion here. And then the tail fibers, I don't know, kind of like an octopus with 6 legs instead of 8 though. Tails are frequently seen in bacterioviruses. By the way, bacterioviruses, we have a specific name for it. It's called bacteriophage. So all that means is bacteriovirus, a virus that infects a bacteria, the domain bacteria. I had one more thing to talk about. There's different proteins in the head than in the sheath and the base plate and the tail fibers. It's only this region here, 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 and there where the attachment molecules of the virus are for attaching to the host cell. Meaning when this virus binds to its host cell, it only attaches at the bottom of the tail fibers. Any question about any of that? So let's move on to viral infection and the host immune response. When a virus infects a host cell that will, assuming that the host cell is an animal, I don't think plants have an immune system, only animals do. It stimulates the host to produce an antibody. Now, obviously, in bacteria, this doesn't happen because they're not animals. But we're talking about when a virus infects an animal cell. I guess I should add that in there. Infection of a virus of an animal cell. That should be added. That's really late on my catching that. This is about 4 or 5 years I've used this slide. Anyways, when a virus infects an animal, it will stimulate the host to produce antibodies. And then the antibodies recognize and bind to the viral surface proteins of the virus. And these viral surface proteins that the antibodies bind to, we call those antigens. Any question about any of that? Some viral proteins can change or mutate frequently, and that will allow different viral strains to infect the same individual more than once in their lifetime. For example, how many of you have had influenza more than once? Come on.
Personally, I have.
Yeah.
I
we
have.
had influenza multiple times in our life. And it's because the influenza virus, it either changes or mutates frequently, and then our body is no longer immune to the virus, because what our immunity fought was the original virus, and now the virus has mutated or changed, and it can infect us again. This is going to be a problem with coronavirus, if you don't know, and that is the virus does change fairly rapidly. And they don't know yet how long the vaccine will last, but there is a very good chance that the virus vaccine for COVID-19 will last 1 year or less. So you may need, with this virus, to get the vaccine more than once a year. And how often we'll need to get it is not well known. And why we know this is because obviously we don't know about COVID-19 yet, but in the coronavirus family of viruses, there are many of them which cause colds in humans, and they're related to COVID-19. And we can get those viruses several times in our lifetime, the same virus, because it changes. Any question about any of that? The flu, of course, was the best example. That one we know. And once you get the vaccine for the flu, 3 years after getting the vaccine, the virus will have changed enough that that vaccine is no longer good, or no longer considered good, because the immunity will be too low. With COVID-19, it'll probably be less than 3 years, but we'll have to find out what it'll be. The flu is a virus that changes very rapidly. Many viruses do not change rapidly. An example, the polio virus. The vaccines were developed in the 1950s, and we're still using those same vaccines to immunize people against polio. So that virus has not changed in, gosh, how many years is that? Let's see, 1960, in 60 years, over 60 years, that virus has not changed. Okay? So some viruses, probably most viruses, do not change rapidly, but some of them do. Any question about any of that?
So do that virus, the polio, will propagate, in a family or not, Professor?
I couldn't hear all of that. Could you get closer to your computer or speak louder?
Sure. I know I'm curious about the polio virus because my grandfather had it, and I'm just wondering if it will transmit, by epigenetics to another generation.
No, it doesn't move from generation to generation. The only chance that he could have spread it was when he actively had a polio infection. He could have spread it to other people who were not vaccinated. Most people in the United States have been vaccinated, although I suspect that because polio is so rare that young children, or maybe even children, a lot of people probably aren't vaccinating their children because polio is fairly rare in the world. To my knowledge, it's only in 2 or 3 countries in the world, and the World Health Organization, and let me think, what the heck is it? The United Nations are trying to make polio extinct in the world. And the 2 or 3 countries where polio is continuing are countries that are unstable and have a lot of fighting, and so it makes it really hard to get in, the health workers to get in and vaccinate the children. I remember Afghanistan, whatever, however you say that, is one of the countries that has polio. And I don't remember, it might be Pakistan is the second country, and I don't remember what the third country was. But in the first world countries, polio has been pretty much extinct for about 20 years. And even 20 years ago, people in the United States generally only got it when they traveled to a country where it was found. So polio hasn't really spread much in the United States and hasn't for over 20 years. All right, let's move on to viral taxonomy. Viruses, we group them into their families. And there are some viruses that are grouped into, is it order? Let me see, King Peter came over from, yeah. So orders, there's a few viruses that are grouped into orders, but generally we don't understand enough about the evolution of viruses because their genome is so small, and then it changes very rapidly to their host, meaning their genome, what's the word, evolves to their host. And so we don't understand the greater groupings of viruses. We just don't follow them, understand them very well. There is one grouping that isn't mentioned by your textbook author, but we do group viruses into the kingdom that they infect. Meaning we talk about bacterial viruses, viruses that infect bacteria cells. We talk about plant viruses, viruses that attack plant cells, and then animal viruses, viruses that infect animal cells. So we do group them by that way. And that probably is evolutionarily related, but that grouping is by the cells that they infect, which is obviously evolutionarily related. So most viruses are only grouped into families, and we group them into families based on their nucleic acid, the sequence of the nucleic acid, the replication strategy, and their morphology. All viral families end in the name Viridae. The genus name of a virus ends in the name virus. And then viral species, that's a group of viruses sharing the same genetic information and the same ecological niche, meaning they infect the same host cells, like COVID-19 is a viral species. Viruses are further categorized by naming them by their common English name. There is no scientific Latin naming system, meaning the polio virus is the common English name. And don't ask me why. Well, I think the reason why English names were used is that the first scientists studying viruses all spoke the English language. And then all later people who started studying viruses continued to use the common English names. Okay? Now, viruses, it's important that we go one step beyond species. And even with bacteria, we will mention that there's different strains or subspecies. Well, subspecies are different in viruses, and we designate subspecies by a number. So we give the viral species name and then give it a number for the subspecies. Let's give it an example. A viral taxonomy. The family is the herpes viruses, like herpes viridae, that's the family. The genus name would be herpes simplex virus, usually just called simplex virus. It does have another common scientific name, which actually is not being adopted by most people, human herpes virus, or human herpes simplex. And then the subspecies would be human herpes virus 1 or herpes simplex virus 1. Okay? The subspecies is important. If you don't know about human herpes simplex 1 that causes cold sores, human herpes simplex 2 is very different in its lesions. It causes genital lesions. Okay? Let me see who's making noise. Somebody want to ask a question?
Yes, I got a question.
Uh-huh. Who's speaking?
Sorry.
Who's speaking? Two people are up.
Oh,
All right, I'm just going to mute both of you. Okay? Now, who's speaking now? So unmute yourself. I had two people unmuted and I was getting background noise, so I wanted to mute. Okay, Courtney, do you want to ask a question?
No.
Okay.
Is my mic on?
No, yeah, you were open before I shut you. Was it Denise? Did you want to ask a question?
yeah. What's about the herpes virus?
I didn't hear the question. Can you repeat that?
Sure. So I want to know if even if it's dormant, can it still be spread?
No, when the herpes virus is dormant, it cannot spread. The trouble is that it can come out of dormancy and then have a minor, very minor lesion where it can spread. And then you wouldn't know that you were capable of spreading it. But obviously, if you have a cold sore for herpes simplex 1, then you know that you can transmit the virus. Okay? But after the cold sore goes away and it goes into dormancy, you cannot transmit the virus. The trouble is, if it comes out and has such a minor sore, you might not realize it, and then you can spread it. And anyways, that's the problem. But my point is that the subspecies of a virus can be very different. Herpes simplex 1 causes a cold sore. Herpes simplex 2 causes a genital lesion. Okay? Another better example showing you how different the subspecies can be is with the poliovirus. And I don't know the numbers for this, but I imagine it's poliovirus 1 is the virus that causes the disease polio. Everybody got that? The second poliovirus is the virus we use for the oral polio vaccine. It does not cause polio, and we use it to vaccinate people to prevent them from getting the polio disease. So that tells you how different the subspecies can be. One gives the disease polio, the other we use as a vaccine to prevent the disease polio. But they are subspecies of the same species of virus. Any question about that? Okay, no questions, let's move on. Growing bacteriophages in the lab. Remember, bacteriophages are just bacteria viruses, and I've actually grown bacteriophages in the lab in this manner shown here. You have to grow all viruses in a host cell. For bacteriophages, you grow them on a lawn of bacteria. A lawn of bacteria is just where you coat the plate so that bacteria grow everywhere on the plate, and that's why it's white everywhere on the plate. And then you put on a solution containing the bacteriophage, spread it around the plate, and then incubate the plate. Then you look at the plate, you'll see plaques in the bacteriophage, lawn of bacteria. So here we have the lawn of bacteria, and there's a plaque. What happens where there's this plaque is there was one virus that attached to a host cell and then replicated in the host cell, lysing the host cell, releasing more viruses, and where do they go? The neighboring cells. And then they infect those cells, replicate, lyse those cells, and then go out and infect more cells. And that's why we see a plaque. All the cells in the plaque, bacteria cells, have been lysed. And then around it, that's where the bacteria are growing. And that's why we see the plaque. Any question about growing bacteriophages in the lab? No questions? Let's move on. Let's, excuse me. Let's talk a little bit more about viral multiplication. The viral genome only contains a few genes, such as the capsid protein genes needed for the new virion synthesis. How a virus multiplies is generally they hijack a host cell and then use that host cell to replicate the virus. Any question about that? So they hijack the cell and then use it to make new virions. Following infections, viruses take over the host cell metabolic machinery and use that host cell machinery to make new virions. It uses the host ribosomes, tRNA, amino acids, ATP, and even some of the host enzymes. And all of these are used to make new virions. The basic mechanism of viral multiplication is similar in all viruses. We're going to be talking about it with bacteriophages, which are sometimes called phages, but I'll use the longer term to make it clear that we're talking about bacterial viruses when I say bacteriophages. Are viruses that infect bacteria cells, and bacteriophages replicate in two mechanisms: the lytic life cycle and the lysogenic life cycle. If a virus follows the lytic life cycle, they infect their host cell, lyse it, and that'll kill the host cell. If the virus follows the lysogenic life cycle, it will infect the host cell, but the host cell will remain alive for as long as the virus stays in the lysogenic life cycle. Any question about these two mechanisms of viral multiplication? All right, so before we talk a little bit further about viral multiplication, I want to ask you, what do we need for a bacteriophage to make new bacteriophages? What do we need? What components does the virus need to make? I didn't hear that answer. Can you repeat it?
Wouldn't it need a host cell?
Yeah, it needs a host cell, but what does the virus need to make in order to replicate the virus? It will use the host cell to make those products, but what do we need to make to replicate a virus? What do all viruses have? Come on, people, what do all viruses have?
DNA.
Yeah, viruses have a genome, and so we need to replicate that genome. If the virus is a double-strand DNA virus, we have to replicate that double-strand DNA to make new copies of double-strand DNA viral genome. And this will occur for whatever the viral genome is, like double-stranded RNA. Well, we still have to replicate it, but it's a little more complex. That's why I'm showing double-strand DNA, because I'm not meaning to be complex here. We just need to replicate the viral genome. We need to then transcribe the viral genome to get viral messenger RNA, and then translate the viral messenger RNA to get viral capsid proteins. And then the capsid proteins have to get together with the viral genome to make the new progeny virions. So this is what all viruses need to replicate themselves. They need to replicate their genome, they need to make their capsid proteins, the capsid proteins need to get together with their viral genome, regardless of what the viral genome is. So let's talk about the lytic life cycle of bacteriophages. Remember, bacteriophages replicate in only two ways, and the first way we're going to talk about is the lytic life cycle. There are 5 stages of the lytic life cycle: attachment, penetration, biosynthesis, maturation, and release. In attachment, the bacteriophage simply attaches to their host cell, which will be a bacteria cell. And then there's penetration, where the phage lysozyme opens up the cell wall. Remember, bacteria have a cell wall, and you have to make a hole in that cell wall in order to infect the cell. And bacteriophages have lysozyme to do that. And then the tail sheath contracts, and that puts pressure on the head, forcing the tail to inject the genome of the virus into the host cell. So that's penetration. The genome of the cell gets inside the bacteria cell. Biosynthesis, the viral genome then makes new phage genome. We're going to make it DNA. And then viral proteins, the capsid proteins. And then there's maturation, assembly of the phage particles. The viral genome gets together with the capsid proteins. And then release. The virus bacteriophage releases the enzyme lysozyme. This lyses the cell wall. It breaks down the cell wall. The host cell will eventually burst without the cell wall protecting the cell membrane, water will come in, and that will eventually cause the cell to burst. And when the cell bursts, it will kill the cell, obviously, and that will release the newly replicated virions. Any question about any of that? If not, let's watch a video. I got it to work this time. I think on this one, I might need to open 2 windows here. Yep. I'm pretty sure the sound doesn't work on this one, so let me get it to open in a second window, and that has the sound but no picture, and that's why I need 2 windows. I know that's really funny. Yeah.
First, the phage particles stick to the surface of the bacterial cell. They bind to
And where's the other one? There we go.
The phage particles stick to the surface of the bacterial cell. They bind to specific sites called phage receptors. Next, the DNA of the phage is injected into the cell, as if from a syringe. The ends join, forming a circular molecule. Then, the injected DNA replicates inside the host bacterial cell, and phage heads and tails are synthesized. The phage DNA is then packaged into the heads, and the filled heads attached to the tails. Finally, the cell dissolves, or lyses, liberating about 100 newly formed phage particles after 45 to 60 minutes.
Yeah, the number of viruses depend on the species, meaning how many viruses are released, and so we won't go into that. All right, let's go up one. Let's come back to my slideshow. Move that over to the side. All right, let's go through viral multiplication, the lytic cycle in a little more detail. First, the virus attaches to the host cell, and remember, it's only the bottom portion of the tail fibers that attach to the host cell, and then the base plate and the pin remove the cell wall in this region, and then the virus contracts, seen here and in there. And that puts pressure on the head, and that injects the viral genome into the host cell, much like a syringe does, injecting fluid into the body. And it's going through the hole that the tail made right there. The cell membrane then reanneals, by the way. And then that's penetration, as simple, so attachment and penetration. Biosynthesis happens. The genome then is transcribed and replicated, and we make new viral genome, as well as the capsid proteins. And in this case, we have several proteins that have to be made. The proteins of the head, the proteins of the tail, and then the proteins we also need to do at the base plate, which isn't shown here, and the tail fibers. The proteins of the tail fiber have to be made. And then you have maturation, where the viral components come together. The viral genome will be put into the viral head. The head will be attached to the sheath, and the sheath attached to the tail fibers. And then the release, where the viral genome, meaning there will be a copy of the virus still in the cell, transcribes lysozyme, which lyses the cell. Okay, any question about any of that? For the lytic life cycle, there's 2 terms you need to know. The burst time, that's the time from the initial infection of the host cell to the time where the cell bursts. And that time will vary depending on the species of the virus as well as the temperature, meaning between 20 and 40 minutes is the average burst time, but at cooler temperatures it takes longer. And I don't know which species would be faster, but some species are faster than others. Okay? And then burst size, that's the number of new virions that are released from the infected host cell, the burst size. And that depends on the species of virus. It's usually between 50 and 200 new virions released in the lytic life cycle, depending on the species. Okay, any questions about any of that? If not, let's move on to the lysogenic life cycle. I think we'll have time to finish this. The second way that bacteriophages can replicate in a bacteria cell is to infect the cell and then enter the lysogenic life cycle. Now, a lytic virus can only perform the lytic life cycle. So a lytic bacteriophage only replicates with the lytic life cycle. But a lysogenic bacteriophage can replicate either using the lysogenic life cycle or the lytic life cycle. So when a lysogenic virus infects a cell, it immediately has to decide which life cycle to enter, the lytic life cycle or the lysogenic life cycle. Once it infects the cell, if it chooses the lytic life cycle, we then have the process of the lytic life cycle, which is the same for a lytic virus. Any question about that? However, if the lysogenic virus decides to enter the lysogenic life cycle, the host cell will remain alive for as long as the virus stays in the lysogenic life cycle. What happens in the lysogenic life cycle, the lysogenic phage, or bacteriophage, sorry, I said I would always use bacteriophage, infects the host cell, decides to follow the lysogenic life cycle, and then the lysogenic virus inserts a copy of its genome, meaning DNA, into the host DNA molecule. And that begins the lysogenic life cycle. The bacteriophage remains inactive, kind of latent. It does not kill the cell, although it can undergo metabolism, so it isn't, well, so can a latent cell or a latent virus. It just doesn't cause disease if it's latent. But the lysogenic virus, when it's inserted into the host DNA, remains inactive and it stays in the lysogenic life cycle. The bacterial host cell carrying a lysogenic virus is termed a lysogenic cell, and the bacteria cell remains alive as long as the virus stays in lysogeny. Any questions about any of that? So let's look at that a little more detail. Once the lysogenic virus infects a cell, it immediately has to decide, do I enter the lytic life cycle or do I enter the lysogenic life cycle? If it enters the lytic life cycle, the virion is replicated, it transcribes messenger RNA and translates that into protein, replicates and matures the viruses. lyses the cell, releasing the virus, which then go on to infect another cell. Once again, when the cell is infected, that virus has to decide to enter the lytic life cycle or the lysogenic life cycle. If the virus decides to enter the lysogenic life cycle, the viral genome integrates into the chromosome of the host cell. This viral genome we call a prophage. That's the part of the DNA of the virus inside the host chromosome. And the only time the prophage replicates is when this cell replicates its DNA. So this cell divides into two. One copy of the chromosome will go into this cell, the other copy will go into that cell, and there will be one prophage replicated in this cell, and then one copy of the prophage in that cell. And this can go on through many cell divisions where the virus is replicating when the chromosome DNA is replicating. Any question about that? Every once in a while, for reasons that are not fully understood, and they've been studying this for over 50 years, I think it's been over 70 years now, the lysogenic prophage can leave the lysogenic life cycle. When it does, the prophage leaves the host chromosome, becomes a separate entity of DNA within the cell. And then that virus enters the lytic life cycle. Okay? So when the virus is in the lysogenic life cycle, it can remain in the lysogenic life cycle. It could do it for many, many generations. Or it might be just one generation. And then it leaves the lysogenic life cycle and enters the lytic life cycle. If it enters the lytic life cycle, then this cell is used to replicate more virions, and the cell will lyse, and that will release the new virions. Any question about any of that? I think we got a little video here. So let's watch that.
It attacks the host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates. and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions about the video? If not, let's move on. We've got a few minutes. Let's see if there's anything I didn't... oh yeah, there's a few things. Let's talk about the lysogenic life cycle in a little more detail. When the lysogenic life cycle, the lysogenic virus genome integrates into the host chromosome, it transcribes a repressor protein, and the function of this repressor protein is to keep the lytic life cycle genes dormant. So I just wanted to say that. And as long as the repressor protein is active, the lysogenic life cycle will continue. But if the repressor protein is not transcribed and translated, or somehow the repressor protein gets knocked off of the genome of the virus, then the lytic life cycle will begin. Any question about that? And then let me reiterate, it isn't well known why the cell or why the virus switches from the lysogenic life cycle to the lytic life cycle. That switch is not well understood, and I'm not going to get into it. I will mention that there is one thing that they do know will cause the switch, and that is if you damage the DNA of this cell, the virus frequently, if not always, will switch to the lytic life cycle, because the virus knows the DNA has been damaged and that this cell may die, and so it enters the lytic life cycle. But other than that, why the switch happens is not well understood. There are a few consequences of lysogeny. One thing that happens, the lysogenic cell is immune to infection by the same bacteriophage. So this cell here, which is lysogenic, cannot get infected by this bacteriophage again. Another consequence of lysogeny is that phage conversion may occur, where the host cell may exhibit new properties from the gene in the bacteriophage. So this cell has a genome and certain properties, and then a virus inserts its genome, and the genome becomes a prophage. We call this a recombinant cell because it now has new genes, new DNA, which can be expressed and change the phenotype of this cell. One example of phage conversion of the cell is Corynebacterium diphtherium. The Corynebacterium diphtherium cell does not produce the Corynebacteria diphtherium toxin unless that cell is infected with the lysogenic virus. The Corynebacterium diphtherium toxin is produced on the lysogenic viral DNA, meaning the prophage. Any question about that? If you don't know, the Corynebacterium diphtherium toxin is a really bad toxin and used to cause major health problems in the United States and throughout the world. It's not seen very much today because of antibiotics. But this is one toxin that the cell itself cannot produce. That cell has to be have a lysogenic virus that has infected it. in order to produce the Corynebacterium diphtherium toxin, because that toxin is encoded by the viral genes. Okay? There's a third consequence of lysogeny. You can read about specialized transduction. We're not going to cover it in the lesson, and you're not going to be tested on it. All right, any question about bacteriophages? Viruses that infect bacteria cells. As I stated, there are viruses which infect every kingdom. We're going to be spending some time talking about the multiplication of animal viruses, because these are the viruses that cause human diseases. Any question about that? The multiplication of animal viruses follows the same basic pattern of the multiplication of bacteriophages, but there are several differences. The attachment, of course, is different because the virus has to attach to the cell membrane, but it still has to attach to the host cell. The penetration is different. You don't have to get through a cell wall and then inject your genome into the cell. Animal viruses usually penetrate the host cell, entry into the host cell, by endocytosis or by fusion. I'll talk a little bit more about both of those later in the next lesson. The encoding can be different, although the encoding can be the same. It can happen by viral or host enzymes. The biosynthesis, generally the same. Maturation, generally the same. The release of animal viruses can be very different. It can be the same, meaning animal viruses can be by rupture of the cell, and that would be similar to the lytic viruses, lysing the cells. But animal viruses can also, or at least some animal viruses, can replicate by the budding life cycle, and actually all enveloped animal viruses replicate by the budding life cycle, and that is totally new that we see in animal cells, and the budding life cycle is not found in bacteriophages. Any question about any of that? Let's talk about this slide very briefly. The animal virus generally follow the same multiplication as bacteriophages, but there are some differences. There's only one that you need to know about, and that is animal viruses can replicate in the nucleus, or they can replicate in the cytoplasm. Bacteriophages can only replicate in the cytoplasm, because that's all there is in bacteria, the cytoplasm. But animal viruses can replicate in the nucleus or in the cytoplasm. And we'll talk a little bit more about that in our next lesson. Are there any question about, well, animal viruses, or any question about bacteria viruses at all? If there's no questions, you can see me from 6:30 to 6:45 if you have questions. If you don't have questions, I'm not going to be lecturing or giving you a lesson or anything. I'm just going to be answering questions. So I'll see you in the lab.
Oh, I'm here to remind you about looking into Lab 8 about Quiz 5.
Yes, I'll do that in between the lecture and the lab. Okay?
Thank you.
All right.